questioning is perhaps the greatest gift we have as human beings.
Science is more than a body of knowledge. It's a way of thinking.
It's a way of thinking that teaches us to question everything we think we know.
Science came about by developing techniques, methodologies for gaining reliable knowledge about the world.
We have at our fingertips the technologies that were only possible for the largest governments and corporations 20 years ago as an individual today.
If the human civilization continued at anything remotely like the current pace of technology advancement for 1 million years, where would we be? I think we're either extinct or on a lot of planets.
The good thing about science is that it's true whether or not you believe in it.
We hold the scientific method in high regard because it works. If it stopped working, we'd throw it out.
Discover the past.
Create the future.
This is the Here and How podcast.
Welcome to the Here and How podcast, where every episode we dive deep into big ideas, exploring the past so that we can create a brighter future. Joining me today are my terrific co-hosts, Stephen Woodford and Rachel Oates. Say hi, guys.
Hi, guys.
So today we're going to be diving into the topic of what is life and where did it come from? So guys, real quick, what is life?
Me.
Rachel, go first.
Oh no, I just meant I am life.
Oh, you're life?
Yeah.
Okay,
So,
you are alive?
yeah, so
And what is it about you that makes you alive?
I'm full of cells that respire and can replicate and stuff like that. And oh my god! I did a thing in school when I was tiny where you have like the 7 signs of a living thing. And it's like reproduction, excretion, respiration, nutrition. I can't remember the rest. There's 3 more. Movement.
Wow.
I think. I might be making that one up.
It's kind of blurry though, because you have some that, are able to do some of those and not others. Steve, what's your definition of life?
I kind of see things as organic and inorganic. So organic is life and inorganic is not. But it really does depend on how you define life.
And how you define organic.
Yeah, exactly. Yeah, it's really definitional, and it's going to places that we, as we're probably going to tap into, we don't know a hell of a lot about. But that's how I would define it. As far as how I would actually put that, I'm not entirely sure, to be honest with you, because if we uploaded a consciousness from a human into a machine, and the consciousness is still there, it can feel pain, etc., that's life. That would change the definition just by itself. So yeah, it's very interesting.
So the historian Fred Speyer defined life as a regime that contains a hereditary program for defining and directing molecular mechanisms that actively extract matter and energy from the environment, with the aid of which matter and energy are converted into building blocks for its own maintenance and, if possible, reproduction. That's kind of a mouthful, but basically he's saying that it's something that is able to have some type of way of pulling energy from the environment and using it for its own survival mechanisms and to reproduce. Where do you think some of the problems with that definition might be?
I see several problems, but the opposite of life is death. So
Is it? Or is the opposite of life just inanimate?
Yeah, but would that be the equivalent of death in the context of defining life in that way?
I don't know, because I don't think of this box as being dead. It's just not living. It's inanimate.
Yes, yeah. It's difficult, isn't it? Yeah.
Mm-hmm.
So
I feel like to be dead, you have to once be alive.
Oh yeah, that's for certain, yeah.
Yeah.
I would agree with that. I wouldn't say death is just being inactive.
Yeah. So my phone
so whatever
is
life
neither
is,
dead or alive, it's just inanimate.
Yeah, because it's never been alive.
Yeah.
So whatever ceases to continue at death, we can call that life, if we can find something that is similar in all things that we consider life. But I actually suspect... Yeah, Karen?
Well, so what I was going to say is one of the problems with that definition is that you have clearly living organisms, like the tardigrade, that you're able to freeze to a point where all their metabolic functions completely stop and cease. And you can rethaw them 10 years later, and they just pop right back up and keep going and are able to survive long periods of inactivity like that. And at the same time, you have organisms like mules that they're not able to reproduce, but no one would deny that a mule is a living creature.
Yeah. It won't surprise me if there's really not a definition that encompasses everything.
Now, I would argue that mules technically, like, there's this fuzzy boundary between life and non-life, and we're going to dive into it throughout this episode. But one thing to really bear in mind is that you are not just one living creature. You are a colony. You are composed of trillions of cells, and each one of those is alive. And there's various levels of that structure where you might have inside your cells, you might have a bunch of different things that are undergoing some form of respiration. And so it's almost like your cell is kind of a cluster of organelles that are, carrying out processes and stuff. And so it's like, where is the fuzzy line between what's alive and what's not? When you die, many of your cells continue to live eating, basically you for a while after.
One thing I'd point out when it comes to like mules and stuff is that they can't reproduce in a sexual sense, in that they can't, produce gametes that go on to create an embryo and another little mule. But they do replicate their own cells, if that makes sense.
Exactly.
In that case, they are kind of reproducing in that way. It's just not sexual reproduction of a new organism.
Yeah, for sure.
I think when we use words like reproduction, we have to kind of specify, are we talking about sexual reproduction, or are we talking about like a replication of cells?
But just before we jump on that, what you were just mentioning there, Thomas, reminds me of the episode that Rachel hosted, which was "What Makes You You." So would it be, as you said, you are a congregation, a collection of organisms, as well as your own conscious entity, or would it be suffice to say that you are the conscious entity? However, you are comprised of entities themselves which are living. It just taps into that area quite nicely.
Yeah. And we're going to dive down into, at kind of the cell level, what makes a cell living and not. One of the definitions given by the National Research Council in 2008, they were trying to determine, when we're looking up into outer space, what should we be looking for when attempting to find life on other planets? And the three requirements that they gave is that it should be in a non-equilibrium state. So it's not just kind of in stasis. It's not just kind of, there's some type of reactions and actions that it's undergoing. It should be in a liquid form, and it should be able to make and break chemical bonds.
So, did you say in a liquid form?
Yeah, and also the first one, wouldn't that necessarily mean that organism you just explained that can be frozen for 10 years doesn't fit that definition?
But I'd kind of argue with that organism. If it's in the frozen state, it's not really living.
It's in a living state.
Yeah, it's just suspended. It's living before and it's living after. But...
So when you sleep, are you alive?
Yes.
Well, yeah, because your cells are still undergoing all kinds of functions. They're breaking and making chemical bonds. They're not in equilibrium.
I see.
There's still subconscious
like
brain
activity. There's like fluid all throughout your body that's being used for these processes.
So it's the movement of organic matter in that sense that you would say that's life. And you would say that organisms that go into a kind of hibernation in which those movements cease or just about cease, that would mean they're no longer alive because those organic bits of matter are no longer moving about. Is that correct? Or
well, that's where it starts getting really tricky. Because a lot of people have this assumption that the line between life and non-life is just extremely binary. It just starts and stops in one location. But there are so many fuzzy areas in between. And the more basic the cells get, there's certain functions that some cells have that others don't. And you can get more and more and more basic and get rid of certain functions and add others and still have particular traits. And it's like, which ones define life? And so we have certain commonalities across most life forms that we can point to. But aside from those, it's like you kind of have to get down to the very, very bare bones to kind of see how life could come from non-life.
Yeah.
And this has been done for a long time, people arguing that, particularly from creationist viewpoints, saying that, oh, scientists would never be able to create life in a lab. Well, depending on how you define life, we either have already done it or are getting pretty damn close. There's a great quote that's attributed to Richard Feynman saying, what I cannot build, I cannot understand. And so scientists are trying to really understand how the cell works by recreating it in a laboratory. A couple examples of this is there's a guy named Martin Hanzik, and he's been experimenting with creating protocells where he'll create in kind of a oil-like membrane. He'll add various elements to this cell and watch it undergo movement, watch it undergo various types of behaviors. It will split off into two cells. It searches out food. And so it's like there's all these different elements. Like it's in a non-equilibrium state, it's in liquid form, and it's able to make and break chemical bonds. So by the National Research Council's definition, that's technically life. Mm-hmm. But a lot of other scientists would look at that and be like, well, okay, but you're not synthesizing DNA. You're not really creating a life form that's able to self-replicate and evolve and grow in the same way that life does.
But a lot of bacteria doesn't synthesize DNA.
Well, but bacteria does possess DNA. And
am I thinking of viruses with just RNA?
Viruses have just
sorry.
Yeah. Even in Martin Hanzik's case, they'd say, well, it doesn't possess any kind of RNA or DNA. It's not able to pass down genetic traits from one generation to the next.
And an analogy that seems to come in quite nicely here, at least I think, tell me if I'm incorrect, is when we're looking at the difference between, say, Homo sapien and Homo erectus, or even further back, we say from Homo sapien to the common ancestor with chimpanzees, Australopithecus afarensis, I think. You can say to people, well, at what point is it a human? And is it Neanderthalensis? Is it Homo habilis? I mean, where is this line? And it seems that each step is important in its own right. One of them is a bigger brain. One of them is upright. One of them is something else. There's just different components. And depending on how you define human, that tells you where that line is.
But even that line is such a blurry gradient.
Of course it is, but it seems like that's the way it is with life as well. So some people would say, and this is a naive, colloquial, informal definition, they say, oh, life is when organisms sexually reproduce. That's a terrible definition, but if you was to define it that way, you have a pretty clear line of where that is. And it's easy to assume that, well, there'll be pretty clear lines in other areas. But as we're diving into, there isn't. It's not as clear cut at all.
Yeah. Well, because you have all these other cells that are reproducing asexually.
Yeah.
Another example of creating life in the lab was done by Craig Venter and his team. And if you're not familiar with Craig Venter, he was the first private company to completely sequence the human genome. And so over the last, I want to say like 15 plus years, they've been trying to create a fully functioning reproducing cell. And in, I think it was 2012, they successfully did this. It had completely synthetic DNA. And he was able to kind of crack down what is the bare minimum that we need. And then they were even able to write things into the DNA, to sign people's names and even add quotes in the language of DNA into a cell that was able to reproduce and function fully autonomously.
That sounds unbelievable. I mean that strictly as that word. That sounds unbelievable.
Now, there's a couple of problems with that when you try to extrapolate that out to the origins of life on Earth, because creating a cell in a lab, you're able to test all kinds of different things, but you're able to do it using synthetic or man-made created enzymes. You're able to completely alter the environment to states that probably didn't exist on the early Earth. And so if we're trying to study how life emerged in, say, 3.8 billion years ago, what we have to do, fossils, we don't have any fossils from that far back because one, most molecules, their molecules aren't going to leave a fossil remnant. They have to have some type of shell or be able to alter their environment in some way, shape, or form. But also the early environment was very, very lots and lots of reactions going on, volcanoes. It was being bombarded by asteroids, and so a lot of those, if there were fossils, would have been destroyed.
Mm.
How old do you guys think the earliest fossils we have are?
I knew this.
It depends on how you define fossil, of course. You're counting just any kind of sign of life, right?
No, specifically fossils. We have stromatolite fossils. How far back do you think those go?
Okay, so I know that we have some kind of indications of life at 4.2 billion years ago, which I only recently learned it's now that far back, which is mental. But as far as fossils, it's hard because I haven't got the right definition in my head for a fossil. It's like 600 million years ago is where lots of them start turning up. But yeah, when in doubt, shut up, eh? So let us know.
You're not gonna venture a guess?
I will venture a guess. I'll say 3.8 billion.
Okay, Rachel.
Billion? Oh, okay. I'm just gonna go conservative, like 700 million years ago.
Okay, so the oldest fossils that we have are actually stromatolites from between 3.5 and 3.7 billion years ago.
Oh,
damn!
And that's earlier than I expected.
What they do is we actually have stromatolites that still exist today, but the fossils, we can kind of compare them and see how these stromatolites are laying down layers of rock and stuff. And we have these rock layers in Australia where we can see the fossil remnants of these stromatolites that date back that far, but they're already basically fully formed colonies, which leaves us an indication that life had been around much longer in order to get to that point. And so when we then go and take large rocks, we may not be able to find fossils from earlier than that, but there are chemical signs of life. So you know how like when we breathe air, we're affecting our environment. We're breathing oxygen out into the environment, and that's affecting the chemical structure of the geosphere, the rocks around us.
Yeah. Plants
are affecting the earth as well. And so we can actually drill down deep into like some of the oldest rocks on Earth, and they've Dr. Ruth Blake of Yale University did this with some rocks in Greenland and found chemical signatures for life as far back as 3.8 billion years ago. I'm not sure where your 4.2 number came from, but as far as I'm aware, 3.8 is the oldest signs of life we've found.
Yeah, I'll quickly Google it to make sure that I'm not giving out false information.
So
it's really cool, though.
Life goes back that far. Now, what I want to make a clear distinction of before we dive into abiogenesis and how it works, I want to make a very, very clear distinction. This is not evolution that we're talking about. We're not talking about once life popped into existence, how it evolved into what it is today. We're not talking about natural selection. We're not talking about evolution. This is a very different topic. This is also very different from how the universe popped into existence. That would be covered by the Big Bang, or where these essential atoms came from in stars. That's stellar nuclear genesis. Once we have a planet, once we have Earth formed, the Earth is going to change. It's going to, its chemical structure is going to change as life emerges. Life and the geosphere, or the rocks, are actually intertwined. There's a link between the geosphere and the biosphere. So, we create chemicals that affect the Earth creates chemicals that affect life. And so they're very, very intertwined. We're not really going to dive into that too much, but we are going to show what we know about how the first cells came about on Earth.
That's such a good point to raise. Quite often when I've spoken about evolution, or somebody who doesn't accept evolution has asked me, can we talk about it? And I'm like, of course we can. Their first question is, well, how did it start? And I have to explain, they're not related. Like, they're linked, but they're not related. It's a bit like if he said or she said, let's talk about gravity. And I went, yep, let's talk about gravity. And they said, well, where does gravity come from? So no, that's not how it works. Gravity is describing how the laws manifest now. It's not telling you about its origin, etc. That's a different thing. So that's a good point. But just to quickly say where I got that from, the wiki entry, which has a timeline on the evolutionary history of life, it's got plenty of resources. It puts single-celled organisms between 3.5 billion and 4.2 billion. So that's where I got that from.
Okay. I think part of that is speculation, just because, before that time, the Earth was extremely hot and extremely tumultuous. As far as I'm aware, the earliest chemical signatures that we actually have that there was some type of life is from 3.8 billion years ago.
Yeah,
but either way, it was a long time ago.
Yeah, I'm fairly certain on that as well. I mean, I haven't looked into it, but it could be speculation, not based on nothing, because it's very hard to get anything to fossilize, let alone things which are tiny in an incredibly rupturous environment 4 billion years ago. You're lucky to have anything.
Well, and even the amount of time that it takes to go from a single-celled organism to a colony like that is huge.
It's by colony. Do you mean multi-cells or?
Multiple cells clustered together, not necessarily one animal or one organism that's multicellular, but several single-cellular organisms working together in some type of symbiotic clump.
Yeah, evidently multicell didn't turn up for, until about, maybe you've got different notes, but I think it's about 1.4 billion years ago. That's a massive wait to go from single-celled to multicelled. And
for the first 2.1 billion years, there were only single-celled prokaryotes.
Yeah,
and those are like the simplest forms of single-cell organisms.
Yeah, as we discussed in our episode on the Drake equation and alien life, that fact there, that it took 2 billion years for multicell to come along, added to the fact that every organism that you test the DNA off, etc., it shows that we all come from one ancestor. Those two facts tell me that life is probably abundant, but that multi-life really isn't.
Yeah. Is now a good time to ask a potentially stupid question or bring up a potentially okay, so you know how like all cells are, like all living cells and stuff are essentially like made up of non-living matter? Right, like where do we draw that line between like what makes something living and not living, like on a cellular level? Like, because we wouldn't say that, like, DNA is living. We wouldn't say that a protein is living. We wouldn't say that, like, an amino acid is living. We wouldn't say that a carbon atom is living. So at what point does it cross that line and suddenly enough things together become living?
Well, and that's kind of what I want to show, is that it's an extremely gradual gradient. And that there's certain functions that we associate with life, but it's not a clear-cut thing at all. So the first point which you made was that everything is chemicals.
Yeah.
We are made up of molecules. We are made up of, all of these things. Like a quick example. Do you know what the four organic chemicals that are found in all living things, you know what they are?
Carbon,
oxygen, hydrogen.
No, more specifically, the four organic molecules would be proteins, nucleic acids, carbohydrates, and lipids.
Yeah, of course.
Yeah. Okay, that's what you mean.
Do you know what amino acids are made out of, or at least the 20 most common ones that are in humans?
I used to have to know all this stuff back when I did biomed.
Hydrogen, oxygen, nitrogen, and carbon.
Yeah.
And there's a couple. The two common amino acids contain sulfur. But do you know what nucleic acids are made out of?
Nucleic.
Go ahead.
Not
hydrogen,
anymore, no.
oxygen, nitrogen, and carbon.
Oh, right. Okay.
If I'm not mistaken,
hydrogen, oxygen, carbon. Like these are all the same building blocks. Lipids, predominantly hydrogen, oxygen, carbon, and sometimes nitrogen or phosphorus. So there's
phospholipids.
with like phospholipids.
Yeah.
But these are all like the same building blocks.
Yeah.
And these building blocks are extremely prevalent throughout the universe.
Yeah, they are.
Like hydrogen, oxygen, some of the most common elements.
Yep.
Yeah.
So
I think they are, aren't they? I thought they are. They are the two most common.
Oxygen is not the second most.
Oh, sorry, no.
There's
helium and
and it depends because this varies between whether it's the most common in our solar system or the most common on planet Earth.
Oh, of course, yeah.
But yeah.
So back in the day when I was studying biomed, I used to have to, I did a whole module on proteins, genes, and genetics, and it was super interesting, but it was really difficult because one of the things we had to do was memorize like every amino acid and its structure. And like, you used to be able to like, he'd shout out an amino acid and you'd like have to draw it and know so many things about it and like where it was found in this and this. And one of the trickiest things was how similar so many of them were, but how big of an impact kind of minor differences in their structure could have.
Yeah.
And that was one of the things that always surprised me but interested me at the same time and made it extremely difficult to remember them all, which I can't do now.
Very true. Yeah. So there's somewhere between a hydrogen atom and a fully functioning human being life emerges. Mm-hmm. Now, in order to really kind of dive into how this field of study has progressed, I want to take a step back in time through history and talk a little bit about vitalism and spontaneous generation. Mm-hmm. Have either of you heard of vitalism before?
Heard of it, don't know much about it.
No, I have heard of spontaneous generation, though.
Yeah.
So vitalism was the idea that every living thing on Earth had some form of vital force in it. And this is very different from the concept of a soul.
Yeah.
Mm-hmm.
Because when you die, they believe that your soul went up into heaven, but that your vital force basically would ooze out of you in the form of like maggots and flies and like give off other forms of like nasty life or whatever.
Yeah, this was some of the ancient Greeks, wasn't it? Like some of them labeled it fire and some of them labeled it air. Is that correct?
I'm not sure about that.
Sure.
But so this idea though that everything had some type of life force, basically they said that you could not create any kind of organic matter from inorganic substances. Until 1828, there was a chemist named Friedrich Wöhler, and he was able to make, can you guess what?
Life.
Was it organic matter from inorganic matter?
He made pee.
Oh,
he synthesized
yeah.
pee. He made it in a bottle completely from just. And so everyone's like, whoa, how did he just made this chemically? Like it was urea, but
that's actually kind of
basically
cool.
he made a bottle of pee and he disproved a theory by making pee.
Ooh.
How do you know how he did that at all? It's crazy.
I don't know the exact chemical process, but I'm sure our listeners could probably look it up.
Awesome, yeah, that's damn.
So
really cool.
the other theory though that needs to be disproven first is the idea of spontaneous generation.
Mm-hmm.
Now, a lot of times creationists will point to this and say, oh, spontaneous generation, it was debunked, way back in the 1800s, that life can't just pop into existence from nothing. But there's a very clear distinction too between spontaneous generation and abiogenesis. Now, this idea goes back as far back as Aristotle, and they believe that there were certain ingredients, like if you put your underwear in a bowl with some wheat, that the next day that mice would spontaneously generate.
Yeah, and I heard a great one, which was essentially it was Greek, ancient Greek. They said if you kill a bull and you put it in a shed and you leave it for a year and you do something else, you'll get bees.
Is this related to when creationists try and do the whole like, oh, if I have this jar, eventually sometime a living thing will appear in it?
Yeah, some maggots or something will appear on top.
Yeah, something like that.
So the first way that scientists tried to disprove this was Francesco Redi. He made an experiment where he put some meat in the bottom of a flask, and one of them he had just kind of a netting over the top of it. One of them he corked it up so that it was airtight, and the other one he had just open air. And the open air container got covered in maggots, but the other ones, like the jar with the net on top, had the maggots forming on top of the net, and the sealed one didn't have anything at all. And so it kind of that was a step, but then they said, oh well, the vital force, it's floating through the air, and if you seal it up, it won't be able to get in. And so Louis Pasteur came up with this hooked flask, and in the neck of the flask, he basically he boiled some water and then examined it for any kind of bacteria. And he found that leaving it, for several weeks or overnight or however long he left it, that the flask was still clean, even though air could get in and technically the vital force should be able to get in too. But there were no bacteria that contaminated it. So he pretty much destroyed the concept of spontaneous generation.
Yeah.
And it was around that time Louis Pasteur came up with the phrase omne vivum ex vivo, or life only comes from life. And then along comes a little old man called Charles Darwin.
Who is that?
I don't know, some guy.
Yeah, doesn't seem important.
Nah, he doesn't seem like the most important person ever.
In a letter to one of his colleagues, he put forth the concept that he said, what if, and oh, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed ready to undergo still more complex changes. Okay, so it's kind of this idea of like a primordial soup that the building blocks of life could emerge through chemical processes. Way ahead of his time.
Yeah, as he was in many areas.
Still purely speculation. There's no evidence for it at this point.
No.
So then another guy comes along, a Russian biochemist, in 1924, and he wrote a book called The Origin of Life. So kind of like The Origin of Species, but in this book, he outlined the chemical stages necessary for life to emerge from non-life. And so his name was Alexander Oparin, and he basically said you'd start with simple molecules, and then those molecules would form into the molecules for life, or he called them biomonomers. They're basically monomers that are organic. And those would form into macromolecules or polymers, or that would form into polymer complexes where they'd all bond together into like strands of DNA and whatnot, metabolic networks, and then a living cell. Still complete speculation. Now, there was one huge experiment that came along in the early 1900s, shortly thereafter. Are you guys familiar with what I might be talking about? It's one of the most famous experiments in all of abiogenesis.
Yes.
Steve?
The Miller-Urey experiment.
Exactly. And Rachel, are you familiar, or are either of you familiar with how this experiment was laid out?
Not a huge amount.
Not so much the layout, but some of the findings they had are just extraordinary.
So what they did, they tried to recreate the water cycle. So like evaporation, condensation, and in that cycle, they put water in the bottom that was heated, and then they put a chemical mixture of gases, like ammonia, and I forget what all else they put in there. I think nitrogen and maybe carbon dioxide. Basically elements that are necessary for life, but that you would expect to find in the early atmosphere of Earth. And they added one key aspect that was a spark. So, you would expect to find lightning, especially if you've ever seen like massive volcanic clouds that create lightning storms inside of them, or, if you have huge thunderstorms, it's pretty common that you'd have some type of spark.
And imagine that the world when it was forming would have been far more aggressive with storms, etc. Just as it would basically be like a mini Jupiter, where there'd just be a massive storm floating around.
Yeah.
But like, of course, whenever you have volcanoes as well, you have all the static and tons of storms there.
Yep.
And so they put this all in a basically they let it run for several days, and they started seeing that the water in the bottom was starting to kind of turn this pinkish color, and around the electrodes there was kind of this dark gooey black stuff that started to form. They left it, but after about a week they tested it, and they found that these simple atoms, or these simple molecules, had formed into, can you guess what?
Amino acids.
Exactly. Amino acids and sugars.
Which is
amazing.
Incredible, yeah.
So completely naturally, they underwent these chemical processes that formed some of the key building blocks for life.
Oh my god.
Now, there's been some criticisms about like, hey, that might not have been exactly what was in the atmosphere. That might not have been exactly how it was in the early stages of the Earth. And so scientists have run even more tests and more simulations.
I think the important thing, though, is that this proves it can be done.
Yep,
exactly.
It doesn't mean it was how it was done, but it proves that it can be done.
Yeah,
so
exactly.
The very first step of Oparin's Origin of Life, simple molecules to the biomonomers, that step was confirmed. Like right there. That was like scientifically tested fact. It can happen, and the Miller-Urey experiment showed it.
That's so amazing. It's so amazing, yeah.
Now, since then,
what
scientists
year was
have
this,
run...
sorry?
What?
Sorry, what year was this?
The Miller-Urey experiment?
Yeah.
It was early 1900s, I want to say like 1940s.
Oh, okay. So kind of
Off the top of
earlier
my
than I
head.
expected.
I could be off on the exact date.
That's okay.
But,
Sorry for interrupting.
Since then, scientists have run simulations of meteor impacts, of volcanic activity, of deep sea underwater vents, and in every single one of these circumstances, they've found the exact same results, like that they've been able to produce amino acids. 24 years after the Miller-Urey experiment, aquanauts, which are basically underwater explorers or scientists, found that in some of the most extreme environments underwater, even as far down as a mile below sea level, that right next to deep sea underwater vents that get up to 600 degrees, that there's life thriving around it completely without sunlight. So life is able to thrive in really extreme circumstances, even caves that are basically covered in like sulfuric acid, which is like battery acid, that there's life thriving in it. So the boundaries for life are much, much broader than we ever thought.
Yeah.
So cool.
So I just wanted to say that one of the most fascinating things I find about that experiment is of the amino acids that they found, they basically created in that experiment amino acids which we found in the earliest dates that we can find any kind of cells, any kind of life whatsoever. So not only did it create amino acids, it created the amino acids that just so happened to be in all organisms as we know it. I thought that's really strong evidence just in and of itself.
This sounds incredible, and just to kind of play devil's advocate, like, what do people use to kind of like argue against this, like in favor of creation or whatever? Like, is there anything you can argue against it?
Yeah.
Well, the most common argument that I hear is the God of the gaps.
Yeah, exactly.
Where they'll point to like specific areas or specific elements where they're like, well, we don't know how this part happened, or we don't know how it went from here to here.
Yeah, yeah.
But those gaps are getting smaller and smaller by the day.
And of people off that mind, there's always going to be gaps, because each time you fill a gap, you actually create two more. So, for example,
not necessarily,
but if you take again humans, the evolution of humans again, as soon as creationists were like, well, where's the gap between a chimp and a Homo sapien? Where is it? And then we found, Australopithecus afarensis. And rather than saying, oh, wow, that is quite interesting, they went, well, now you've got two gaps. So you find Homo habilis, and they go, oh, now you've got three gaps. There comes a point where it just gets really stupid.
Until you have every single generation.
Yeah, exactly.
There's the Futurama sketch, though, isn't there? where Farnsworth keeps, like, bringing out new, like, skeletons and species, and like, what came between them?
I haven't seen that. That's great.
Oh, it's so good.
Yeah.
Can we link it in the description?
Turtles all the way down.
So we've been able to simulate some of these other environments. One awesome one was we've actually found meteorites from outer space that have contained amino acids and sugars inside of them, and so there's a theory that maybe there's somewhere in our solar system that's even better at creating these, or that's an even better has a better atmosphere or better environment for creating these building blocks for life. And maybe it traveled to Earth, like it got knocked into our in the direction of our planet and was carried to Earth on some type of meteor.
That on it in and of itself is absolutely incredible again, but it actually brings to my mind a question regarding the Drake equation, essentially the episode which we did a while back, and that is, if that counts as life according to definitions, that means we have found life that that is not...
The amino acids themselves are not defined as life yet.
I see. Would some definitions of life say it is because it's got biological movement, or that doesn't count as life at all?
No, it doesn't. It doesn't have any kind of autonomous movement. It's not self-replicating yet. There's no cell membrane, and those are additional steps that we'll cover.
Yeah.
But one really cool aspect is, so this isn't a living organism that's able to survive a meteor impact. These are just amino acids. They're just the building blocks right now. It's like making Lego pieces.
Sure.
Now, Dr. Jennifer Blanc simulated a meteor impact in a high-pressure cannon that was basically firing it at such intense speeds you'd think that everything would just be pulverized and destroyed. But what she actually found was that the amino acids not only survived, but they fused into more complex peptides.
That's cool.
Which are they're now ready to make RNA or DNA. This is insane. And so, and it's important to point out just how many amino acids they found. They found between 70 and 100 plus just from meteorites.
Wow.
Scientists simulating volcanoes have found that there's one experiment by Bob Hazen where he simulated deep sea vents, and initially he came up with no amino acids, but then he was like, okay, well, what if they required some of the other elements that would be around them, like some of the rocks and minerals that they're situated next to? And when he ground those up and added it to the experiment, sure enough, amino acids resulted.
I had no idea we actually had that much evidence. I thought so much of it was just speculation and theories, but the fact that people have actually done simulations of this is incredible to me.
Yeah,
this is
I'm in the same boat
all
as
around the world by thousands of scientists in hundreds of different institutes and labs.
It's amazing.
It's nuts just how much research you have to sift through.
I'm quite gobsmacked, really. I didn't realize we have just this much evidence, just a scratch in the surface, it seems. I think partly it's because a lot of people very much care, why are organisms the way they are? I very much care about that question, and that's why evolution has been so important in my life, because it's just revealed basically so many answers. But I've never really personally cared how life got here, or how it originally originated, because evolution was in and of itself enough for me to really put aside any kind of belief in some kind of God or a purpose in that kind. And so the origin of life itself has just never been a super interesting. It's definitely interesting, but never been a concern of mine. So I've almost bought into the assumptions and the sayings that we don't actually know a hell of a lot about abiogenesis. It seems that we absolutely do. It's incredible.
I can't help but feel that if we kind of spoke about this more as a society, it might turn a lot more people away from creationism and that kind of thinking. Because one of their big arguments is that, you don't really know how old fossils are, even though we do. Or, there's a gap here, or this, or we've never seen evolution, that kind of thing. But these are literally simulations where we've seen evidence of the theories and what we expect.
Oh, it gets even cooler. I'm just scratching the surface.
See, I think this is the kind of stuff that we need to be talking about on a big, wide scale and making sure everyone knows about and gets excited about, because it's amazing.
I'm definitely going to take my research from this and make a video on Holy Kool-Aid about it.
You should, for sure.
Oh, I'd watch
condense it down.
Yeah.
Yeah. Something just to go on that, the showing these kind of experiments a bit more. You're absolutely right, Rachel. Most creationists that I've met, they respond much better to observational science than they do to, I forget the right term for it, but it's when you're...
Which is kind of ironic, really, because...
Yeah, I know. Well, they respond bad to both, otherwise they wouldn't be a creationist, but they respond better to observation than they do to... I'm forgetting the word right now, but it's when you basically...
theoretical stuff?
Forensic, that's the one.
Oh, okay.
So, for example, you can know with real certainty that somebody committed a crime by the evidence that's left behind. Do you know it with certainty? No, of course you don't. But I reject certainty in that kind of definition outright. It's easier if you just see it, if there's observation. But there is evidence enough to do it. It's like cutting a tree down.
That's the thing, though. All of this is forensics. You're using chemistry and geology to find, like, okay, if you stumble across a crime scene and there's a body, you're pretty sure that somebody died who.
Yeah. So if somebody says to you,
if you find a fossil, you're literally finding a body.
Yeah.
You know, if you're able to chemically analyze it and analyze the DNA structure of it and stuff, you're performing forensics.
No, that's exactly it. But if for example, you made the claim we can create amino acids, and I said no, we can't, an experiment which I can observe really would shut me down. But if you was to say this tree that we've just found that's been knocked down, it started life 10,000 years ago. A lot of people will just say, well, you don't know that. And they're shut off because, again, it goes to the forensic nature of it. They don't understand how you can have a very accurate position statement that's based on very good evidence to make that case. So yeah, there's the difference if you see where I'm coming from on that point.
I think a lot of it is just a misunderstanding of how a lot of this works.
That reminds me of I guess kind of like a debate that Ken Ham and Bill Nye did. And when Ken Ham was showing Bill Nye around, the Ark encounter, and they came to a bit where they were looking at snowfall from the Arctic, and they did the thing where, you get like a really thin sheet, and you can see the layers of snowfall, and from that you can kind of extrapolate back how old it is, how far it went back, when each layer of snowfall fell, and stuff like that. And Bill Nye was trying to explain all of this and be like, this is what it means, this is how we know this, and we can do these tests. And Ken Ham was like, well, that's silly. How do we know all that didn't just fall overnight? And it was like,
yeah,
it just reminds me of that.
No, it does. That's exactly how it works.
The ice core analysis.
Yes, yeah.
Yeah. That's exactly how it works. It's you can't prove it in that sense. It's like, that's not how it works. The evidence indicates this. But yeah.
Well, for the sake of time, I'm going to push through some of this a little more.
Yeah, of course.
One of the next big steps is like you can have all of this, all of these amino acids, but they still have to form together to make strands of RNA or DNA, and they have to be enclosed in some type of cell membrane. So the cell membrane, there's been a lot of breakthroughs. Dr. Jack Sozak at Harvard University has done a lot of research on it. There's other scientists as well who've found that if you can have a cell membrane form pretty naturally from fatty acids. And while we once thought that fatty acids were only formed by living organisms, if you put like hydrogen and carbon monoxide and you heat them together in combination with certain minerals that are found in the Earth's crust, then they form fatty acids completely naturally on their own without needing any kind of living cells. And if this happens in like underground pressurized chambers, where you have like heat from magma and stuff, it can easily accomplish this. So like they're simulating these environments that we have on Earth, all over the Earth, that would have existed way back, at the dawn of time on Earth. And what gets crazier is as pressure forces some of these molecules up into like pools of water, they naturally start to cluster together. And so you have, one end of them is hydrophobic, and one end is not. And so they basically form where the hydrophobic part is centered in, and they form spherical-like structures. Yeah,
and that's how we get like phospholipid bilayers, because like you say, it looks like quite a complex structure when you like see it or examine it, but really it's just kind of common sense that it would form that kind of structure because these parts like to be near water and these parts don't, so they just cluster together. And
when we say when you're saying they just cluster together, I guess what you're just saying is the way they behave under the laws or under physical laws is that's just what they do. Because some people would hear that and go, well, what's making them do it? And it's like, well, they don't. It's just that's how they react under
bonds.
that.
Yeah.
They're chemical
I don't
bonds, and we can
humanize
observe
them
this.
too much, but they're just going into the state where they're most comfortable.
Yeah, exactly. Yeah, no, I was actually referring to the language Thomas used, but yeah, for sure.
But I mean, all of this, like all of these chemical processes, it's like there's certain atoms that have a spare electron and some that don't, and they bond together. These are just the forces of nature that are driving this.
Yeah,
it's not some type of spooky magic that we don't know how it works. Like this is very, very this is high school level chemistry.
Yeah, exactly. Yeah. So is that amino acids we now can demonstrate can be created under natural processes, and so can lipids.
Can become trapped inside of cell membranes.
Wow.
Now, because these cell membranes, they're very primitive at this point. It's not like a modern cell wall yet, but they're semi-permeable, so you can have these amino acids entering and exiting through their cell wall. And so if they're in a dense enough concentration, then these amino acids can start to accumulate inside of them. And at this point, these amino acids can start to form together. And, if you have, we showed naturally they can make peptides as well. And as these peptides, form together, you can start to get them pairing together into strands of RNA. Now, most of us, all of us, all of our cellular mechanisms are encoded in DNA, but we have RNA as well. If you start to get more basic, you look at viruses. Viruses only have RNA, not DNA. And an RNA is a simple single strand. It's not double-stranded. It's just an open-ended strand. But there's some theories that put forth basically an RNA world hypothesis, where before DNA, RNA is what was used to pass down genetic traits heritably from one generation to the next. The reasons that we think that there's it was an RNA world rather than a DNA world is that, one, the RNA base pairs bond together, forming a single strand naturally. The replication process is really simple. If they're in kind of like a soup of other amino acids, then the amino acids that connect to them, if you remember, you have 4 base pairs with RNA. Those, it's, what is it? Uracil, adenine,
thymine,
and cytosine.
Oh, cytosine.
I think thymine is
thymine is the one that's replaced with
uracil.
Yeah, that's it. Yeah, sorry.
So you have these base pairs that they naturally connect to each other, but not to the other base pairs. And so if you're sitting in kind of this soup of base pairs, then the connecting points, the puzzle pieces or Lego pieces will come together and click into place. And suddenly you have a double strand of RNA where all these pieces are coming together and lining up on it. And all you need is a little bit of heat and they'll split apart. And then you have a duplicate pair. Now, the problem, we run into a couple of issues that are a little difficult for scientists to replicate. One of them is that the self-assembly process is kind of difficult. The backbone atoms of RNA don't form chemical bonds with each other without some assistance from lab technicians, some type of enzyme or something that will cause them to bond together easier. But this gets kind of cool because there's one, first of all, in the presence of iron and the absence of oxygen, which in the early Earth, it wasn't a heavily oxidized environment. There weren't plants that were spewing oxygen out into the atmosphere. And iron was very common throughout the oceans. We can tell this because if you dig down enough in the Earth's crust, there's a particular layer of iron deposits where iron basically interacted with, or iron in like the oceans interacted with oxygen that was being pumped up by algae, and it formed these bonds that then caused it to sink to the bottom and create iron deposits. Wow. But if you take an environment that has no oxygen and a heavy concentration of iron, then RNA starts behaving differently. And so we can simulate this. And at the Georgia Institute of Technology, they simulated this, and they found that RNA became an instruction carrier like DNA, and it carried out even an electron transfer, which is necessary to jumpstart reactions like photosynthesis and respiration. So it starts behaving in completely different ways. Another experiment that was done was... So in this case, they're altering the environment that RNA is in, but sometimes scientists will actually alter the RNA itself slightly. So like Dr. Nicholas Hudd and the Center for Chemical Evolution, they changed the components of RNA by just 1 or 2 atoms, and all of a sudden it reacted much easier, stacked naturally together, and sometimes thousands or even hundreds of thousands of molecules bonded together. So it became much, much more stable. And so this kind of leads to this theory of maybe before RNA, there was Proto-RNA, and that all it would take is a slight mutation then for it to change into strands of RNA or even DNA.
That's
Damn.
amazing.
Yeah, that wow. Mind blown a little bit, to be honest. How strong is that connection or that hypothesis or theory of RNA becoming DNA? Do you happen to know if there's a decent amount of evidence for that, or is that what we suspect at the present with this relatively small amount of evidence that we have?
So, I mean, these are only a couple studies that I'm mentioning. There are hundreds of people who have been performing experiments on this, and it seems to be the most viable hypothesis right now.
Damn, that's wow.
Or I should say theory, not hypothesis, just because of the amount of testing that's been done that seems to confirm it.
Yeah. So, just to go back to the definition of life, at what point is it life? So it's not single-celled. Is that right? Sorry, it's not amino acid because it doesn't move. Is it when it's in a cell? Is it when RNA appears, DNA appears, or is it completely not related to these definitions at all? It's somewhere else.
I think the cell membrane is a big stepping stone towards whether it's life or not, but I don't know if it's the...
Well, cell membranes, I mean, we've found come together pretty naturally. Amino acids come together naturally. Proto-RNA comes together naturally. Here's where it gets cool, and this is kind of the boundary that I would say kind of makes it more lifelike, is ribozymes. So essentially, if you have a strand of RNA that's laid out flat, and there's not enough amino acids around it to come and bind to it, or not enough base pairs to come and connect to it and create a replica, the strand at times can actually fold back on itself and connect to the appropriate connecting base pairs. When this happens, it creates kind of this loop, and the base pairs that don't connect face outward. When they do this, they can actually perform certain functions. And so these ribozymes will start either joining together or breaking apart certain molecules around them in their environment. And so now you have a naturally occurring, naturally forming ribozyme that's able to carry out certain reactions in its environment. And here's where I think it becomes lifelike. Some of the functions can give it a survival advantage. Let's say, for example, that it's able to create from molecules, that it's able to create nucleotides, okay, which are the building blocks for DNA and RNA. Then if it's able to create more of those, then it could create an environment that's even better for creating more RNA and replicating itself even faster. Well, scientists at Simon Fraser University, they examined hundreds of strands of RNA chains, and they found several of them that were able to form ribozymes capable of building nucleotides, but they did it really poorly. So these ones, they're doing it kind of, in a crappy, haphazard way, but they selected out the most successful ones, and then they replicated them using PCR, and just like you would expect in the environment that eventually, they'd replicate and you get more and more of them.
Just speeding up the natural process.
Just speeding it up so we don't have to wait millions of years. And they found after just 10 generations of mutation and selection, that's nothing, considering that like certain bacteria will multiply like thousands of times in a day. Or I'm assuming that number, I forget the exact, but they can replicate at massive rates.
Yeah.
10 generations isn't that long, especially in the terms of billions of years. After just 10 generations, they found ribozymes that became extremely proficient at creating nucleotides. So at this point, what you have, this isn't biological evolution, this is chemical evolution. But it's happening completely naturally with organic molecules forming something that's able to sustain itself better. At this point, the line between life and non-life is, as far as I'm concerned, it's as close to crossing it as you can get.
Yeah, I would agree with that.
We can go a step further. When you have something like this, it's very, very simple, right? You have a simple cell wall. Well, okay, at this point you have ribozymes that are able to break down certain molecules. Maybe you have these that are reproducing, and suddenly you have, millions of them all across the earth, and, they're encountering other ones that are similar, and they're breaking them down, and some of them are surviving, some of them are not. Well, let's say that you have a particular cell that, it's starting to kind of get really, really good at this, and it becomes, some type of protist, some really simple cell or protobiont. If, let's say, it's able to consume another cell, but it's not able to break it down or digest it, then suddenly it has within its cell wall, it has another cell living inside of it. This could be advantageous. And so scientists have actually observed this happening with various protists that will, eat some type of algae or something, not be able to digest it. And then all of a sudden they have this other organism living inside of it that is undergoing functions that are beneficial to the cell itself. So in this case, this would be a perfect example of where our mitochondria came about.
Yeah.
Mitochondria, they're organelles living inside of our cells that are completely separate, they're undergoing their own processes that are creating power or food for us. Their waste products are our food.
Yeah.
And so this theory is called endosymbiosis, and it's basically how you get from these simple protocells, how you get more complex structures inside of them that are symbiotically working together to create a more complex prokaryote.
So I think mitochondria is probably like the obvious example that a lot of people know about, but are there any other examples?
I think plasmids.
Oh, really?
Would be. I'm not sure too much. It's
know
been a
about
while
that
since I've looked heavily into the anatomy of modern cells.
Yeah. No, that's okay. Mitochondria was the example we were given at uni as well, so I didn't know if there were any others that were well known.
So at this point you have more complex cells emerging and developing new traits and new features and characteristics that might be beneficial. Now, let's say that one of them develops a new tool and it develops the ability to move slowly through water, some type of flagellum or something. Now, this cell is able to engulf other cells and eat them up and use their molecules as food or as resources to propagate itself throughout, or replicate itself and propagate itself throughout the environment. It's going to survive better. But if it encounters a cluster of cells, let's say that it comes across, and this has also been observed with protists. I think this was protists and algae with endosymbiosis. I think I said algae. I'm not sure if that's correct. But essentially, if let's say that a protist comes across a couple of algae molecules that when they were separating, if they had a difficult time separating and they're clustered together, it becomes more difficult for the protist to consume it or to eat it. And so there's kind of this advantageous symbiotic relationship that forms between algae molecules where the ones that clump together are more likely to survive. And so you start seeing these clumps and these clusters arise after just a few generations. And that's kind of the origin of having multicellular colonies. Now, if these colonies start to behave in ways where they survive better together and they reproduce together, then and eventually they become dependent on each other, and some of them are not able to reproduce on their own, or they're not able to survive on their own, then you start to see the emergence of multicellular organisms. And at this point, things are starting to get much more complex. The components of the cells are getting much more complex. The DNA strands are getting longer. They're having more functions. And so you start seeing these complex aspects of cells emerge, and then you have hundreds of millions of years of evolution kind of tweaking and adding and stuff. You have the arising of DNA and other aspects, but kind of getting to this point is where I want to stop for this episode just because of time constraints. But it kind of shows you how you can go from just basic hydrogen and oxygen and carbon and nitrogen, just basic atoms and elements, all the way up to these molecules that are forming naturally, or up into, amino acids and RNA strands and cell membranes. And obviously cells are much, much more complex now because they've had a lot of time to get there. But to just assume that, like, to take a snapshot of, like, right now and be like, oh, look, it's so perfect. How could it possibly pop into existence out of nowhere?
Yeah.
Spontaneous generation was disproved. It's like, no, we're not talking about spontaneous generation. We're not talking about vitalism. We're talking about this really gradual change over time.
It's so simple when you explain it in those steps as well, though. Like, it can be really overwhelming to try and think about, like, oh, how do we go from, like, you know, one single atom to a human being? Like, that seems overwhelming and scary. But when you explain it like you just did, with, like, step by step by step, and then take into account, you know, like, the evolution from multicellular organisms all the way up to what we have today, it just seems so simple. All it takes is time.
Yeah, so we know that with natural selection, right? With evolution by natural selection. To those who are privy to the evidence and who has really looked into it, it's really not hard to conceive how the most simple of replicating forms can go all the way to human and much further, because it's not a ladder, of course. It's just it's easy because natural selection explains exactly why that would happen. The most interesting thing to me seems to be in relation to abiogenesis, that is, when is the baton really passed from chemical evolution to biological evolution? For me personally, at the present, it seems to be when something is replicating. What stage is that exactly?
I mean,
yeah, at what point?
Theoretically, it can happen. And probably does happen as early as when you have your nucleotides inside of the right cell membrane.
And is
they
replication
start bonding together and replicating?
Yeah. So is replication when something brings in energy, or however you want to define it, other matter, other atoms, into itself and then produces a copy of itself? That would be replication, right?
That would be self-replication,
okay,
because it's producing a copy of itself.
Whereas,
if in the absence of some type of enzyme to do it, it would require some type of chemical process to cause it to split apart. So we know if you have a strand of RNA and the nucleotides are all lining up and pairing with it, then you have two, connected strands. Without some type of enzyme to break it apart, you would need some type of chemical process to do that naturally, whether it's, some type of heat or something that, causes them to pull apart. But you would still need some type of natural chemical process.
I see. Because in my mind, that is the most pivotal point. Once that's nailed down, as far as I'm pretty much concerned, abiogenesis is done with, which is to say a lot. But maybe I'm thinking about that incorrectly. Do you think it would be wrong of me to be putting so much emphasis on that one point there?
Well, I think that is one of the most crucial points, just because once you have the ability to self-replicate your strands of RNA can undergo mutations. And those mutations can give it a survival advantage.
Yeah.
And so you already have, natural selection then playing a part on chemical evolution. So where it goes from there, I mean, there's a million different routes it can branch out from, but and you can say, oh, it's not really alive, it doesn't have, particular functions or something, but that is definitely a pivotal point in the process of creating life from non-life.
Well, damn, you made me very thirsty to learn more, and I hope the audience has the same impression. You've blown my mind a couple of times throughout this. I genuinely did not know that we knew so much about abiogenesis in general. And yeah, as I said, I'm definitely interested. So I'm going to go away and do a lot of research myself. So thanks for that, Thomas. That's
I
incredible.
feel a little bit in awe right now. Like I'm so impressed with people. Oh, it's amazing. Like the fact that
incredible.
Yeah, the fact we've discovered so much, and like the fact we've done the simulations and seen the results, I think it's incredible.
Yeah, I mean, it's not conclusive, if it doesn't seem. Oh, some of them are, for sure. Some statements are absolutely conclusive. So if somebody says the building blocks of life, and they say the building blocks of life are amino acids, they say that can't occur, let's say, without some kind of intelligence. Well, we know that it's absolutely wrong. So there's certain statements and really important statements that are just that's no longer a thing. That's debunked. That's flawed. There might be other areas where we don't know our answers too well with hypotheses, etc. But that's incredible. It really is a lot of things to think about.
Yeah.
I think there may have been a couple points in this recording where I referred to amino acids or nucleic acids as amino acids.
How dare you?
Just looking back on my notes, and I just want to clarify that. Both can come about naturally. But nucleic acids are the bases for like DNA and RNA, and amino acids are what make proteins.
You can always pop some notes over the video.
Yeah. That's yeah.
I'm always worried as I make these because I'm just flying through a massive amount of notes, and I tend to accidentally mix words every once in a while.
Yeah.
And for that, I apologize, and I encourage all of our listeners to make corrections. Like if I say something wrong, let me know, and
I think
I'll try to correct it next time.
I think
it's human error. It happens.
Yeah, the other thing that's gonna that comes with the territory. We've decided to have this podcast to be a little bit more of a conversation rather than a lecture. Particularly mine and Thomas's YouTube channels tend to be very much fact checked, etc. Whereas here it's a conversation about really interesting topics. So it's bound to occur where we make the odd mistake and we express our opinion.
Well, I want to point out that I'm not a scientist. I'm a science enthusiast. And so while the people who are undergoing this research have dedicated decades of their life to really study the ins and outs of all this and are doing it, we're blowing through a big idea every single week. And a week's time is just not enough time to give these topics their due credit or to really thoroughly understand all of it. But I'm, breaking it down as best I can given that.
Yeah, no, I mean, sometimes it is actually really easy to get your head around something, but there is millions and millions of hours investment. If you take Charles Darwin's natural selection, for example, that took an enormous amount of time. It took an enormous amount of resources. But once the theory's in place, you can explain it very, very quickly. And you can actually demonstrate it just logically, let alone through evidence. Although I say you should show it through evidence as well. So often we're the lucky enthusiasts just explaining what these very courageous and hardworking people have managed to find by thinking very critically about these really difficult questions.
Yeah. The other caveat I want to make is that while each of these steps I'm kind of breaking down as simplistically I can for the sake of understanding, that doesn't mean that that the chemistry behind it is easy to comprehend or that there isn't much more to dive into. So while every single day, there's new breakthroughs and discovering new aspects of where we came from, the complexities that it requires to get us there. So for example, I mentioned Craig Venter and his team, creating a fully synthetic or fully functioning cell with synthetic DNA. They had to go through hundreds and hundreds and hundreds of genes trying to figure out which ones were absolutely essential and which ones weren't, and then go down even further, But even though they're coming up with something as simple as they possibly can, that doesn't mean that the simplest cells that we know of today that have evolved to what they are today, that there aren't immense levels of complexity that have layered on top of layers of complexity to get us there.
Yeah, I mean, that's exactly the case. That's definitely a good point to end on. But I actually want to end, even though it's your episode, I think a better place to end would be, according to how you define life, Thomas, has the question of abiogenesis been answered?
I think that there's still a lot of gaps, but that said, the gaps are more in the exact mechanisms rather than did it happen or not. And the reason that I say that is if you have a gigantic puzzle and you fill up a ton of the pieces and you can see kind of what the picture is, but you're missing some of them, you still have a pretty good idea of what the picture is. And if it is a picture of a castle and you have all the pieces, make it look like a picture of a castle, you're not going to suddenly be like, oh, I don't think that that's a castle. That's actually a cat.
Exactly.
We have enough pieces in place where to me, it's pretty much confirmed that this is what happened. The exact mechanisms and what the exact missing pieces are, we haven't found some of them, but it's pretty conclusive to me that, life did arise from no life.
Yeah.
And we're learning more and more about exactly how that happened every day.
So someone was to say, how do you know, do we know exactly how life arose? The answer would be no, but we have suspicions. But if someone was to say, do we know whether or not life can come from non-life, you would say that the evidence is strongly in favor of saying yes, it can. Yeah, that's great. I think I'm with you as well. Mind blown.
Mm-hmm.
Rachel, did I convince you as well?
Oh yes. Sorry, I'm just sat here kind of wowed. I'm really excited to go away and read more about this now.
Yeah, same.
That's awesome. Then my goal here is complete. I want all of our listeners as well to leave with said enthusiasm. And I'm going to end on a quote real quick, but just remember that next week, Rachel will be presenting an episode on why we find things cute.
It's a very serious topic.
Yeah, we're
practically cancer curing.
We're getting real heavy next week.
So Lord Kelvin, who was a creationist, said, "If a probable solution consistent with the ordinary course of nature can be found, we must not invoke an abnormal act of creative power." Carl Sagan later wrote in a foreword to Stephen Hawking's book, A Brief History of Time, "A universe with no edges in space, no beginning or end in time, and nothing for a creator to do." Once we put all the pieces together, what's the point in then trying to hand off the baton to an unnecessary entity?
Yes. I love that.
This has been an absolute blast, and now we want you to join in the conversation over on the Here and How Facebook and Twitter pages, or follow us on Pinterest and Instagram if you just want some of the dankest of science-y memes. If you like one of our particular styles, check out each of our YouTube channels: Rachel Oates, Rationality Rules by Stephen Woodford, and Holy Kool-Aid by me, Thomas Westbrook. To find all of our episodes, show notes, contact information, and more, warp on over to our home base, thehereandhow.com. If you enjoyed this episode and want us to succeed in spreading the love of science, you can help us out by giving us a 5-star review on iTunes or Stitcher. And to all our friends and family listening, thank you for spending this episode with us. We'll be back to explore another exciting big idea next week. Now go create something magnificent.